Hey all, welcome to ShareTrek. This is Raj here. Friends, Dr. Jeff Boyle is the Chief Scientific Officer of American Gene Technologies. Jeff has over 25 years of experience in immunology, research, assay developments, and commercialization, and has held leadership positions at biopharmaceutical and diagnostic companies. Dr. Boyle has also maintained a long research career with over 30 publications and has received multiple high level academic awards. He's an inventor listed on several patents in the field, including vaccines, immuno-oncology, and medical devices that have been launched as revenue-generating products globally. He has broad experience in responding to examiners, opposition proceedings, and has participated in the defense and prosecution of a variety of IP assets. He has also managed IP portfolios and executed on strategic initiatives to develop and in-license IP necessary for product development as well as market protection. The scope of Dr. Boyle's research and patent work includes diagnostics, utilizing the optimal stimulation of CD4 and CD8 T cells, and ability to differentiate and use simple analytical techniques, diagnostic utilizing the stimulation of both innate and adaptive immune responses to assess immune function, peptide epitopes for T cell based diagnostics, antigen targeting to sites of immune induction, including DNA vaccines, and combination of biologics and immune stimulatory complexes for oncology therapies. I had the opportunity today to talk to Dr. Boyle about AGT 103-T and get his insights. I hope you enjoy our interview with Dr. Boyle. Let's get started. Hey all, welcome to ShareTrek. We have Dr. Jeff Boyle from American Gene Technologies with us today. We are fortunate to have some time from him, and we're going to ask the questions that you have been asking me. And welcome, Dr. Boyle.
Yeah, thank you. Great to be here.
Yeah, so we'll go into the set of questions that I had. The first thing is that we wanted to understand that AGT 103-T is optimized to target HIV-1, which is the top priority because HIV-1 accounts for almost 95% of all infections. Are we looking at anything specific for HIV-2 at a later date?
Yeah, well, I mean, HIV, getting HIV-1 done will be a pretty big accomplishment, and I mean, we're early in that process, and we also have a number of different approaches, sort of in the background that should we need to find further improvements to manufacturing, cost of goods, etc. So our focus will be pretty much on getting HIV-1 done and dusted, and then also our focus is very much on the US to begin with, and then Europe, but then it's a big world out there, and we'll have to see how applicable our approach is for the broader global community, and what modifications may or may not be needed down the track. So that'll be our focus. It'll be, there's a lot to continue to do. So once we get that done and dusted, then obviously we can look to how do we expand it even further.
And fortunately, HIV-1 is almost 95% of the infection. And yeah. HIV-2 is very localized, I believe. So this is really optimum for the community. The other question I had was that in our earlier interviews with Jeff, he had described how AGT 103-T works. Now that we know how it targets CCR5 and includes short interrupting RNA to interrupt viral RNA, can you please expand further if it's possible? Let's say you extract CD4 T cells from a patient. Some of it may be pristine and some of it infected. How do you handle them? Any extra information you can give will be very interesting to us.
Yeah, sure. So maybe start at the beginning. So, our approach really comes down to the concept is that we take CD4 T cells, HIV-specific CD4 T cells, and then we sort of form a bulkhead of protected immune cells and allow those to then, once they're back reinfused into the body, to be the sort of the helpers that then are able to, provide the needed signals to help with really the mechanisms by which HIV is controlled, which are things like CD8 T cells, potentially antibody, although it's never really been shown to be that critical, but that said, it could very well play a role. So those CD4 T cells are the bastion from which the rest of it can adapt with the immune with the virus as an ongoing chronic viral infection. And so in terms of then how we go about doing that, when we start with the patient's own cells, they're infected, they're on drugs, they're well controlled, so they have very low to no virus detected, but there's a low level of ongoing viral replication that leads to a lot of the complications associated with HIV, the chronic inflammation, et cetera. But that's also gives an underlying immune response that's there. So we start with that. It's very, very small, and we take all the cells out, and the way that we target those is through what cells we activate. So we have some synthetic molecules that mimic HIV, and so we activate those cells with those synthetic peptides, and that then makes them susceptible. The activated cells makes them susceptible to infection with our lentiviral vector, and in that lentiviral vector is how we're trying to protect them. And we do that at two different levels. The first one is, so there'll be a population of these HIV cells that were not infected. And so we want to knock down CCR5. So that's going to be your main, protection from infection once you've pulled these people off drugs. And then as a backup, so should they already be infected, or that they get infected by a different mechanism, then we target two areas of the HIV genome. HIV is incredibly diverse, and it has a super high mutation rate, and so trying to find regions of HIV that are so it's called conservation, and the reason they're conserved is because they're coding for regions that HIV uses over and over again, and for multiple different proteins. So there's a lot of selection pressure on those regions to not change. So we target a couple of those, and then any RNA that's produced that contains those targets gets degraded through the silencing mechanisms. So that's the sort of the backup strategy, and we use two different ones so that there's again more pressure applied to stop these mutations from being able to escape.
So to summarize, I think once you harvest the cells from the patients, you are separating out the ones which are not infected, doing a CCR5 suppression. And then you're also introducing mechanisms inside those cells so that if HIV were to get into the cell through some other mechanism like CXCR4 or something, then I think you're targeting the highly conserved areas of HIV, which do not change because that's vital for replication and construction of the virus. Probably you're talking about the trimer or GAG protein, something like that, which is highly conserved. And then because of that, the replication cannot happen inside, even if it somehow manages to sneak into the CD4 T cell. And this empowered T cell is then put back into the patient, and that will stay in the patient's body and mobilize CD8 and helper cells in order to continue the functional cure on the patient. Is that a reasonable understanding?
Yeah, and I think the big thing is that we don't actually separate out the infected versus uninfected.
Okay.
I mean, it's actually very difficult to do that because one of the challenges with HIV is that it's can be latent. So it's integrated and it's doing nothing. So there's nothing really to identify those cells at all, even if you wanted to. and then the other side to it is that we're targeting the HIV specific CD4 T cells via the stimulation with the GAG. We use GAG peptides, as you were right there, and that's a very highly conserved area that we use to activate those cells. So we know that there'll be a good bulkhead from which to provide an ongoing helper response. And then, so once we stimulate them, we then we get rid of all the non-CD4 cells by depletion. So we look at different cocktails of antibodies to get rid of the other cells. So we're really now focusing on the CD4 cells. Then we put our viral vector in there, protect them, and then there's an expansion period for about a week in which we then massively expand the numbers of these cells. So we try to get at least 1 billion of these cells, but it varies depending on patient to patient, what you're starting with and what you end up with.
And I think that because you're putting the interrupting molecule inside, even though you do not differentiate between HIV and non-HIV infected cells, I think it will still have an inhibitionary effect on the replication of the embedded provirus in those CD4 T cells. So I think that's how it would work, I suppose.
Yeah, exactly. So if they were infected and they tried to start to actively, make new viruses, the silencing mechanisms will delete that RNA.
Right. And how do these reinfused CD4, modified CD4 T cells manage to remain in the body for the longer term? Do they multiply by themselves, or what's the mechanisms involved in there?
So it's similar to, you just think about like a typical chronic infection, of which people live with a lot of different chronic infections. You sort of then you set up really homeostatic mechanism between your immune system and the virus. So the virus produces antigens that, as it replicates, that stimulate the immune system gets on top of it, suppresses the virus down, and it's just this ebb and flow then forever. And so you have a certain number of those cells. When they're activated, they become, what's called like effector cells. So those are the ones that go out and actually, suppress the virus. They stimulate CD8 T cells, which kill infected cells, and that drops the virus down. And so the cells, drop down. Some of those cells become memory cells, and those, live in the bone marrow, and at future dates when the virus comes back, some of those get reactivated, and then they become the next ones, the next cells that generate the effector cells. So it's an ongoing self-renewal process.
Okay.
That's again requiring the virus to generate its own ongoing immune response.
Right. So the memory cells do the magic to a good extent. Yeah.
Yeah, they're your long-term reservoir of the next generation of cells.
Got it. And the other thing I had in mind was that I think it makes total sense to target HIV-1, given it accounts for 95% of the infection. I think we touched upon these questions earlier before, but the only tweak I would do here is it possible for you to also shut off CXCR4 at a later date, or it's a more challenging target?
So the main if you look at, say, natural resistance to HIV, CCR5 is a very, it's critical, and people that have mutations in it are naturally resistant, and most of the infection is really driven through CCR5. All HIV, has the potential to switch to CXCR4, and mixed infections. It's more common in, say, untreated HIV, when this chronic infection, you start to see the rise of this alternative. So that is something that we don't anticipate is necessary in terms of the long term, but but in the meantime, that's really why we have the backup of targeting HIV directly, just in case this
Inside the
does
cell.
require.
Yeah, got
Exactly.
it. That's a more elegant solution. Can you please provide us any updates on the progress of phase 1B or phase 2 trial, and when it will be started, if you have any information?
Yeah, so I mean, we're really, right now we're working through, the spin out of the company. Our protocol is currently, we've submitted it to the FDA. So we'll see if there are any comments back from it. We have our sites selected, and so it'll just be a matter of getting through the AdImmune spin out and all of that associated activities, and then we'll be ready to go and then go as fast as we can.
Yeah. And I suppose that you would like to have a statistically significant data available at the end of your next phase. So you might be having a lot more patients in the clinical trial.
Yeah, so when we learned a lot from our phase 1 study. I mean, it was primarily a safety study, as they are, and then with the data that we were able to generate during the ATI, we learned a lot, but we didn't learn everything. And so our first port of call for the next study is really to understand at what point should we take people off their drugs relative to infusion. And so, the first, 3 arms of the study are really to look at optimum timing around that. We saw something quite unexpected in the first study, and when we looked at, the patients as they remained on drugs. We saw that our cells expanded after infusion. We really anticipated, these are well-controlled patients on drugs, so we didn't expect there to be much HIV stimulating themselves once they got reinfused, and yet they expanded. And so that peaked at around 2 weeks after infusion, and then they sort of did more what you would expect to see, absent them seeing virus, and start to fall away. So the next study will really be trying to understand how do we get the optimum response relative to that expansion, and when they see the virus, because you take them off drugs and it can take up to a month before that virus really rebounds in patients. So there's this timing element, and so that's our first real question. And so and we have both the cell therapy side to this and the gene therapy side. So looking at, what impact do those cells have, the number, and what impact does, the immune response itself have. So we can look at other studies that have been done in the past, and where they've just taken people off drugs and multiple times, and look at what you would expect in terms of seeing, control. So we have them powered enough to tell us that, yeah, we're likely, if we get above a certain proportion of people that are able to control virus below that long-term non-progressive level, out for a period of about 3 or 4 months, that we're seeing something, that we're really confident that this is something different. And so we have those groups powered enough to show us, if each of us, we have 3 groups with different timings, and so we can tell, with a good degree of confidence that we're seeing something different. And then, once we see that, then that's really where we then, we take that data, go back to the FDA and say, hey, we think we're seeing something here. We think this is our best timing for infusion versus taking people withdrawing their drugs. And we really want to now, agree on what is it that you would accept as long-term efficacy, durable efficacy, and expand the groups once we get that data. And obviously, no more safety concerns arise.
Can you
I hope you
please
don't.
describe the process of administering AGT 103-T to patients? I believe it's an autologous process. Would you have designated collection centers, just like what these gene therapy companies are doing for sickle cell disease and TDT?
Sure. Yeah. Yeah. I mean, the beauty of what we're doing is that, we don't have to reinvent the wheel here. There's, the steps that we're using to make this product are well established. And because of the nature of the product, because we're using what would be closer to what a natural immune response is looking like, we don't anticipate a lot of the issues that you see when you're trying to sort of get around the normal immune mechanisms, like where you're trying to take the brakes off the immune response. Those are the areas where you're really starting to run into much higher risks of things like cytokine release syndrome, where these cells are sort of get out of control. So everything that we're doing on the patient side can be done in an outpatient setting, which is tremendous.
Right.
So they don't have to be hospitalized like a lot of the other procedures. We don't need to do ablation or reduce their bone marrow and such. So
Right.
we're really just using some differentiated CD4 T cells. So on that side, from the patient side, they can do it in an outpatient setting. From a manufacturing side, we still need to get those cells because we don't want them to lose their function. From the point from you take them out of the patient's body, you really want to get them into the manufacturing process as quickly as possible. So they'll still need to be, as close to the manufacturing site as you can make it, so that you don't run into logistical issues of shipping. And then it's a 12-day process to do the manufacturing, and then the cells are frozen, and then it really takes from the point that they're frozen, it really gives us time to QC test them, and that's really to make sure that there was no issues with contamination of the product, and it's they're sterile, and there's nothing dangerous that's been introduced during the process, and then there's some aspects to do with gene therapy, making sure that we haven't introduced a new replication competent virus into the patients that could be a danger. That's really what takes the longest period of time to test that. And so on average, it takes about 3 months by the time you send these things out to specialist labs for testing to get the results back. And then you've got to, make sure that the patient's fine and schedule them back to come back to get these frozen cells shipped to the site and reinfused. And, so that's currently the process. And so there's a lot of labor in that, it's all about scalability. As you bring these up, you can automate more, you get more, value out of your facility and the people that you're hiring. So the notion would be to really, try to keep these things as centralized as you can, so that you can make the most out of your capacity build.
Yeah, that sounds like a logical approach. You want to put a balance between proximity to the patient, as well as centralizing the manufacturing. Find an optimum balance wherever possible to keep your break-even costs low, so that it can be value for the patient as well as profitable for the company to go further. So that sounds great. So I think everything looks great, and all the ShareTrek community is rooting for AGT 103-T to succeed. They're anxiously waiting for phase 2 to start and to get some data and other things. Can you just briefly give us an idea of how long do you think the phase 2 will be? Like just a rough timeline to manage the expectations.
Yeah, so we're looking at probably a couple of years. Really is a realistic time frame for how long it would take to to finish the next study. And then I said it's really depends on how our goal is to expand at least one of these groups into be a much larger group, and then it really comes down to recruiting. I think the good thing about the HIV is that there's a lot of highly motivated individuals out there that are willing to really commit to what these clinical trials require, which is a lot of visits, a lot of blood donations. So it's a huge ask on the patients, but that's usually one of the things that's time will be the time constraints on how quickly you can get these done is how many people out there are willing to sacrifice what's required in order to further these things along and get the data that we need.
Our community of viewers with HIV come not only from the US but from across the world, and especially the ones in the US. Some of them would be definitely happy to volunteer, because I keep on getting some questions. I tell them you have to go to AGT's website and register, and then they will contact you. Is that the way it will continue to work, given that AdImmune has been created?
Yeah, so I mean, we'll be hands off on these studies. So but I think down the track, once our sites are open and recruiting, these things are all there's clinicaltrials.gov and our sites, which are independent, will be will be the best place for these people to really contact. But we'll make all of that very as public as we can.
Thanks a lot, Dr. Jeff Boyle, and it's been a wonderful conversation with you, and I hope our audience has got most of the things that they were looking for, and we hope to see you again, and we wish you the very best in your endeavors.
Yep, terrific. No, it's happy to talk.
Well, friends, that was an interesting and refreshing chat I had with Dr. Boyle. I hope you guys got most of your frequently asked questions answered about AGT 103-T. I guess that given the state of flux that the company is in, with AdImmune coming into place and transferring the assets from American Gene Technologies to AdImmune, it's not always possible to give too much details given the dynamic nature of the business and research. We will get more and more information as time goes by and in subsequent interviews with AGT. And I'm sure that as and when they have more information to release, they will get in touch with us. So please let me know your thoughts and questions in the comments below. I'll accumulate them for the next time we meet with AGT. Thanks and have a great day. Bye for now.